In last video, I have shown how to use haplotype caller to call variants for one sample, the mother's BAM file. And this video, let's joint call 3 samples together. We still use the same tool, GATK haplotype caller, and the same reference. And this time we use the 3 BAM files in the BAMs folder, mother, father, and the son for the trial. And the output file, we name it trialhc.vcf, and we still use the genomic interval that we are interested in. Here, I make sure I'm in the correct folder and copy-paste the command. Now it's finished. Let's look at the VCF output file in the Geno browser, IGV. Okay, now I already load the trial HC VCF file. We can look at the 3 individuals one by one. Mother is NA12A7A. If we click the variant bar for the mother, it shows the genotype information for her is GTT 2-base insertion and the GTTT 3-base insertion. This is the same as what we have seen for the realigned BAM file. And the type is the heterozygous variants. And let's look at for the father, NA12A77. At the same location, the genotype for father is G, the reference, and the GTT, the 2 bases insertion. And let's look at the son. For the son, the genotype is homozygous variant. Its GTT is both of the chromosome has 2 bases insertion. One allele is from father, and one allele is from mother. If we look at the trial HC variant bar, it will show the allele frequency. Here, the allele frequency is 0.667 and 0.167. The alternate allele has 2 bases insertion, which is more frequent, and the 3 base insertion is less frequent. After variant call, we usually do a filtering step, and there are 2 filtering approach. The easiest one is hard filtering. It's just use some threshold, and this is applicable to all the dataset, but requires some expertise to define it appropriately. And the second approach is variant quality score recalibration using machine learning. It's also called VQSR. Please note this is different from the BQSR, which is base quality score recalibration in the data pre-processing step. This is in the data post-processing step. After we got those variants, we use the variant quality score to do the recalibration. This one is more powerful than the hard filtering, but it requires well-known dataset for the training data. So for human, we can have very good resource, but for other species, I'm not sure if we can have quality training dataset. So in this computer lab, we'll just use the basic hard filtering, and we set a threshold QD. And let's look at what is QD. Let's look at how many variants in each VCF file. Okay, there are over 400 in the mother haplotype caller VCF and only 34 in the final trial HC VCF. So let's look at this mother HC file. Let's search what is QD mean. Okay, here in the header section, it's noted QD is a variant confidence or the quality by depth. Okay, let's filter the mother hc.vcf file by this QD threshold. Okay, now we have the filtered VCF file. So in the filtered VCF file, it's has 3 more lines than the mother HC VCF file. That is because it only mark whether it's passed the filter or not, but it will not remove the variance. And in addition to that, it will add some lines to describe what is the filter name for this particular file. Now let's go to the filtered VCF file. So here is the filter name. This one is added automatically for the low quality filter, and the ID QD10 is the filter I named it, and the description is the QD score is less than 10. And now let's look at the variant part of this file. Most of them pass this filter, but this variant failed the QD10 filter, same as this variant, and the same as this variant call.